Hi students, this is your lecture number 13 in this course, which is Biology 2 online. And this lecture is about the immune system, or the immunity, the system that defends you from harm, from invaders, from diseases, etc. So let's take a look at what is it. Immune system is a system of cells, tissues, organs, reactions, etc., which guard against potentially or actually foreign, potentially or actually harmful substances which are foreign. Right? You never know who is this foreign agent. Maybe a spy, maybe a saboteur or something. So when you, for example, inhale a particle of dust, you have no idea what microbes are on this particle of dust. Or when you step on a rusty nail or something. So again, there is some invasion of certain foreign substances which may be very, very bad for you. And so this immune system that defends you from these invaders can be classified into two categories. First of all, we have innate or natural immunity, a system of so-called non-specific responses. It's a system of organs, parts of organs, substances and reactions which do not discriminate between different invaders. If it is invading you from outside, if it is a foreigner, just destroy it, get rid of it. And elements of the innate immunity or natural immunity or non-specific responses, they don't care what kind of microbe is it. Is it a bacterium or virus or something else? What kind of bacterium? What kind of virus? What they know is that it is not me, it is not the host, it is not my element, part of me, so I'll destroy it. So, for example, there is a cell called macrophage which participates in this non-specific responses. When the macrophage encounters a foreign particle, it engulfs it. Remember, we talked about phagocytosis, amoebas phagocytose, some white blood cells phagocytose, macrophages do this phagocytosis. And the only thing the macrophage cares about is that this is a stranger, this is a foreign agent, right? It doesn't matter what kind. Uh, and this natural immunity or non-specific responses can be imagined as two lines of defense. Imagine a football field, and there's one line of defense, and there's another line of defense closer to the goal. The first line of defense is what an intruder encounters first: skin, mucous membranes or mucosal surfaces. And it says here normal flora. I would change it to normal microbiota, a population of microorganisms which already live in you, are actually helpful. For example, there are some bacteria that produce vitamins in your large intestine. And these guys they don't like intruders. They will develop a certain response and eliminate. Then second line of defense, innate immune cells, which are macrophages, several kinds of white blood cells, granulocytes, eosinophils, basophils, etc. Then it's inflammation. It's not a thing. It's rather a process. It is a reaction, a non-specific reaction to an intruder, invader. Then complement, also second line of defense. Complement is a mixture of proteins which do harm to bacteria. We will have a more closer look at it when we come to that point. And then antimicrobial substances. Mucosal surfaces are first line of defense because when you inhale something, for example, it sticks to the mucosa of the respiratory tract. But this mucosa of the respiratory tract also has microbicidal substances like lysozyme. Also, there are microbicidal substances that kill microorganisms in your saliva, in your sweat, and in your tears. And so all these substances are not, again, non-specific. They don't care what kind of foreign agent is it. They just uh, interact with it, and if it is foreign, not part of me, they destroy it. And then there is a third line of defense. And you see it's a different rectangle, and it says adaptive or acquired immunity or specific responses. That includes, it says specialized lymphocytes. Lymphocytes are actually the main players in this game of adaptive or acquired immunity or specific responses. They are specialized. Uh, each lymphocyte has its own favorite or unfavorite, whatever you call it, selected foreign intruder called antigen. Lymphocyte John only cares about destruction of influenza virus, and lymphocyte Jane only cares about the destruction of a bacterium that causes, say, diarrhea. They are both lymphocytes, John and Jane, but they have their own specialty. Right? They don't share their responsibilities. If you show influenza virus to lymphocyte Jane, she will not react to it. She will ignore it. If you show this diarrhea-causing bacterium to lymphocyte John, again, it will, he, John is he, right? This lymphocyte will ignore this antigen. It will not do anything. It's not specific for just this lymphocyte. These specialized lymphocytes are divided into two large categories. We will talk about that later. B cells or B lymphocytes, cells that produce antibodies. Again, more about antibodies in the lecture. And T cells. T cells are also lymphocytes. They are specific to certain antigens, but they don't produce antibodies. They deal with antigen using some other means, and we will not detail these means. You will learn all the details if you enroll in the immunology course, which actually I am teaching usually. This is my research background. I've been a postdoctoral fellow in laboratories that do research in immunology, so I love to teach immunology. So you will learn details about what exactly T cells do. It's too much for this introductory course. And the T cells also, they are subdivided into two categories: helper T cells and killer T cells. We will talk about their differences. And there's also a third category, the so-called regulatory T cells. But more about that later. Now, just the take-home message. There are non-specific responses which are performed by first line of defense and second line of defense. The first line of defense are barriers like skin, mucosal surfaces, membranes, and normal microbiota. And the second line of defense is what follows this original first encounter, or when the invader kind of compromises, breaks through this first line of defense. Then there are innate immune cells like macrophages, or the so-called natural killer cells and other. And there is inflammation. It's a reaction to an intruder, a non-specific reaction. Then there is complement. It's a mixture of proteins in the blood. And there are microbicidal or antimicrobial substances. Now, if that fails, then we have a third line of defense, which is adaptive or acquired immunity or specific responses, and it is the task of specialized lymphocytes to participate in this third line of defense. By the way, why adaptive? Because the specific responses, immune responses, can change with time. Innate immune mechanisms do not change. We are born with them. When we are born, our body has genes which are responsible for the synthesis of a protein that we call lysozyme, and it will be secreted by several types of cells, and it will be always the same. But that does not apply to lymphocytes. They adapt to different conditions. They change with time. For example, B lymphocytes, which produce antibodies, with time they can produce better antibodies. They sort of learn how to better deal with the intruder. So that's adaptive immune system. Then this is basically the same slide, but shown in a sequence, barriers to invasion. So imagine that there is a foreign material, or we say foreign antigen. It attacks you, and it encounters the first line of defense. Barriers, skin, mucous membranes. Stomach acid, by the way, also wonderful barrier. Stomach acid kills most bacteria, only very few bacteria survive it. Lysozyme, etc. And also mechanisms of expulsion, coughing, sneezing, vomiting, diarrhea. We usually do not talk about them in discussions about immunity, but yeah, well, they have a certain role in eliminating or expulsion of the intruder antigen. Then we have second line of defense, which is certain cells like macrophages, neutrophils, eosinophils, and we have the complement system. And finally, we have the third line of defense, which is adaptive immunity or acquired immunity, and it is mediated by cells, lymphocytes. It is very specialized in that one lymphocyte knows only its own specific antigen, and it can be cellular or humoral. Humoral, the Latin word humor, actually doesn't mean humor in our understanding of this word. It means liquid or body fluid. Right? So B lymphocytes produce antibodies. Antibodies circulate in body fluids. So we say antibodies are responsible for humoral immunity. And cellular immunity is mediated by cells, more specifically by cytotoxic or cell-destroying cytotoxic T lymphocytes. Then let's take a little bit closer look. We're not going to really discuss all the in and outs, all the names of different cells that participate in immunity, all the substances, but we'll just take a little bit closer look at all these three lines of defense. First line of defense, barriers. Example, normal skin. It's a wonderful barrier. You see, your skin has the outer layer or upper layer called epidermis, upper in this figure. It consists of cells which are very rich in water-insoluble protein called keratin. It's the same protein that is used by the organism to make hairs and to make nails. And so it's sturdy and not soluble in water. Hardly any intruder can break through these millions of cells filled with keratin. Well, still, intruders find gaps, but see, there is a lipid mortar between keratin-containing cells. It's made of lipids. Lipids are, again, they are difficult to break through because lipids do not allow water-soluble substances to cross their layers. Oil and water do not mix. Still, intruders do find some ways to enter. They find like nicks, cuts, erosions on the skin, and usually they go through there because these parts of the skin do not have much of healthy keratin-containing cells and lipid mortar. Then the underlining layers of the skin, notice that they contain green colored in the slide cells which have kind of appendages called dendrites. They are dendritic cells, and look, where did they come from? From the bottom layer of the skin, basal or base layer of the skin. And they can move actually through the layers up, that are above them. They're very motile moving cells. These cells are part of the immune system. They have a name uh, honoring the uh, German scientist who first described them, Langerhans, or they are called dendritic cells because of these dendrite-like appendages. They are very active. They eat, swallow foreign particles, internalize them, then they bring them back to the base layer and then to the underlying layers, to the connective tissue, where they present pieces of the swallowed antigens to lymphocytes, and that actually develops immune, begins a specific immune response. Mucosal surfaces also very nice barrier. This slide shows your upper respiratory tract and trachea. They are lined up by mucus, right? There is mucus in your nose, there is mucus in your larynx, there is mucus in your trachea, and also similar mucus is in the gastrointestinal tract, in the alimentary canal, and similar mucus is also in the female genital tract, in the vagina and in the cervix of the uterus. The respiratory system has one unique feature. On the mucosal surfaces of the respiratory tract, there are special appendages called cilia, and they move rhythmically from up to down, from up to up, down, up, down, up, down. And they are sort of driving particles that you inhale to the mucus and make these particles stick to the mucus. You do not really appreciate the role of these cilia until, God forbid, you or, again, God forbid, your child becomes ill with pertussis, whooping cough. The bacterium that causes pertussis paralyzes the cilia. Cilia stop moving, and under this condition, the inhaled particles go down to the primary and secondary bronchi in the lung. And this causes this very strong coughing reflex. And so you better get vaccinated and also vaccinate your kids against pertussis. It's not a nice thing. Right? In other parts of the or other systems that have mucosal surfaces, there are no moving cilia, but there are these goblet cells shown here which produce mucus. And mucus traps foreign particles and also destroys microorganisms because mucus contains microbicidal substances. Then second line of defense is innate cells that are responsible for non-specific immune responses and various chemicals. And the slide details what chemicals are defending you from intruders. Tears contain lysozyme. Mucus, already talked about it. Saliva has lots of lysozyme and other substances that kill microbes. Sweat is very microbicidal. Then more detailed about lysozyme, it is an enzyme actually found in many secretions, including what is written above, tears, mucus, saliva, etc. It is an enzyme that catalyzes the hydrolysis or splitting of molecules in the cell walls of bacteria. So it's a powerful means against bacterial invasion. Then there is a substance called lactate or lactic acid. The sweat is rich in lactate, and lactate slows down bacterial growth. Very important. So in addition to lysozyme, which breaks microbes, it cannot break all of them. There are billions of them if you get an infection. But lactate slows down their growth. So whatever is not killed by lysozyme, it is not allowed to proliferate by lactate in sweat. And gastric juice, hydrogen chloride, the pH, if you recall pH from Biology 1 or from basic chemistry course, the pH in most of your secretions and also in blood is about 7, more exactly 7.3, 7.35, and it is close to neutral. 7 is exactly neutral pH. But the pH in the gastric juice inside the stomach is 1 to 1.5, right? So what's the difference in the number of free hydrogen ions between, say, a sample of saliva and a sample of gastric juice? 7 versus 1, right? It's 6 orders of magnitude. So gastric juice contains 1 million times more free hydrogen ions than saliva or and the liquid that is in the intestine, or the liquid that is in the respiratory tract, or plasma of your blood. Right? So these conditions, when there are so many hydrogen ions, they're very unfriendly to bacteria and other pathogens. They kill mercilessly. Very few microbes actually survive this harsh treatment in the stomach. When we talked about the lines of defense, we mentioned inflammation. It's a non-specific response to invasion of microbes and other potentially or actually dangerous things. When you get a nick or a cut and you don't treat it, next day you see it's inflamed. Right? The area of the cut is red, swollen, and painful by touch. Right? So these are classic symptoms of inflammation. And also the local temperature in the area of inflammation is higher than in neighboring tissues, also a classic sign of inflammation. So what happens during inflammation? You see a wound here, and some foreign invaders intrude. Platelets rush to the wound and stimulate blood clotting. Right? So there is a clot in the wound, which actually is microbicidal by itself. Then a number of types of white blood cells, neutrophils especially, and also mast cells and macrophages. Macrophages are products of maturation of monocytes in the blood. They are microbicidal. In what way? They produce the so-called mediators of inflammation. You see these punctuated blue lines with arrows that come from neutrophils. They symbolize these mediators of inflammation. The most common mediator of inflammation is histamine, but there are others. Serotonin, you probably heard about it, like it's a substance produced by neurons causing happiness. But serotonin also helps with inflammation. And there are others. There is, I think, the last most recent list of mediators of inflammation is 60 different substances. All of them have principally the same activities. They slow down the flow of blood by dilating blood capillaries. Right? This creates redness and increase in the local temperature. Then also, when they continue to slow down the flow of blood, the blood plasma, which is actually liquid, right? It oozes from the capillaries into the tissues, right? Escapes from the circulation into the tissues, causing swelling. Then these mediators of inflammation trigger special receptors of pain, right? So inflamed sites are usually painful by touch, and if a surgeon operates on an inflamed tissue, surgeon always needs local anesthesia, like Novocaine or lidocaine, etc. Then macrophages. Macrophages produce also these mediators of inflammation, and also they produce cytokines. These are substances that are signaling to cells within the circulatory system to do various things, and particularly during inflammation, they signal to cells in the inflammation, to white blood cells, to exit the blood vessel, blood capillary especially, and rush to the site of inflammation, continue to produce these inflammatory mediators, and eventually creating very uncomfortable environment for the intruder. You see, when it is swollen, so there is a lot of bactericidal substances from the blood plasma at the site of inflammation. Then it is hot, relatively, a higher local temperature. This slows down the division of bacteria and other pathogens. And it has a bigger acidity, and these mediators of inflammation make pH of the site of inflammation to go down, right? So they create local acidities. All these are unfriendly, harsh substances for an invader, and essentially they lead to death and expulsion of this invader. When inflammatory reactions go too far, they can be also harmful for the host, for you, right? Inflammation, like a boil or carbuncle on the skin, or inflammation in the sinuses, or inflammation in the tonsils, etc. It can create a lot of discomfort. It needs to be treated. But when it is at moderation, it is not very harmful to you. It is actually more harmful to invaders. So we view it as a defense reaction, by part of these lines of defense of the non-specific immune reaction. Now, we mentioned complement, and I said complement is a mixture of proteins. Right? Note that it is spent with E after L, not I after L, so it's not complement. Right? You may compliment my lecture. You can give me compliments for the lecture, but when you write complement, remember that it is different. It has this AE, not I, after the L. And because it is a mixture of different proteins, sometimes students get this message that it should be written in plural, and they say, write in the exam, complements do this and this and this. That's not correct. It is a mixture, but it is a mixture of proteins that work together. So it is usually, this word is in singular. Complement. We say complement is, complement does. Complement is one thing, although it's a mixture. And what exactly does it do? It assembles the individual proteins in this mixture of proteins in the blood plasma. They, or serum. Serum is basically blood plasma minus fibrin, so it's pretty much close to blood plasma. These proteins, individual proteins, come together and assemble into something that reminds you of a dagger or a sword. And this dagger attacks the membrane of foreign cells, especially bacterial cells, but also other foreign cells. And you see here complement in action. It has drilled a hole in the bacterial membrane. So the extracellular fluids, water particularly, water rushes in, different solutions from outside rush in. So the bacterial cells become swollen because of water, and also part of the solution, some of the solutions that are in the extracellular fluid but move into bacterial cells contain a lot of calcium, and that's lethal. Excess of calcium is like a lethal injection for a bacterial cell, so it dies. And also proteins of complement assist in inflammation. Remember the previous slide? And in blood clotting. I said that blood clot has its own role in the defense. So the complement proteins assist in the forming of the blood clot, and they continue to act as microbicidal agents in the clot. Third line of defense, specific immune responses or adapted immunity. They are mediated by lymphocytes. You see, on the left, you see a picture of lymphocyte among red blood cells. It's not a very large cell. It is larger than red blood cells, but not very large. And it looks kind of dull. There is nothing really in it. Or you don't see anything using simple microscopy and staining. You see a almost perfectly rounded cell. This distinguishes these cells from macrophages, which have an irregular shape, or dendritic cells, which have dendrites or an irregular shape. But lymphocytes are always almost ideally spherical, rounded. Very thin rim of cytoplasm, so in old times, histologists, morphologists, what's there to study in the lymphocyte? It doesn't even have a cytoplasm. And a large nucleus which occupies almost the entire cell. Right? But everything changed in our understanding of lymphocytes when biologists began to look at the surface of lymphocytes. The surface of any lymphocytes contains millions of different substances that stick out like antennae. And because of these antennas, lymphocytes are part of the specific immune system, is that they recognize different kinds of foreign invaders or antigens. Their surface structures are very different in shape, right? And they accommodate different antigens. We'll talk about it a little bit later. These antennas on the surface of B cells can be what we call antibodies, right? And these antibodies can be detached from the surface of the B lymphocyte and circulate in body fluids, and they are specific to particular antigens. They chase them and bind them and contribute into their destruction and elimination. Now, T cells do not produce antibodies. You see a T cell to the right, and it has a red colored thing on its surface. It's definitely distinct from antibodies on the surface of B cells. So T lymphocytes deal with antigens, also specific antigens, because you see this red stuff also has something in it which can be different and accommodate different antigens. But using some other means, not producing antibodies. But T lymphocytes are as specific, as important, as deadly to foreign invaders as B lymphocytes. The favorite target for T cells is viruses and tumor cells. Why is that? Because T lymphocytes are surveying the surfaces of our own cells and find cells which have markers of foreign stuff. Like if a cell is infected with the virus, there are some viral products on its surface, and a T lymphocyte detects these foreign things on the surface of normally our cells, and develops a response. Also, same thing, tumor cells. Tumors, it's not the right notion. We are used to this notion that tumors are something that only old people have. Actually, all of us develop tumors almost from the time of birth, very young age, but the immune system, particularly T lymphocytes, do not allow these tumor cells to divide, to grow and produce real tumors, because, again, they see markers of malignization, of the markers of the process that makes normal cell cancerous cell, and they destroy cells, our own cells, if they show markers of tumor. The T cells are mercilessly destroyed. Then a little bit about innate and acquired immunity again. See, I like the slide in that it kind of visually shows you what innate immune mechanisms do and what specific or adaptive immunity does. On the top of this uh, page, you have a drawing. It's a cartoon. It's not really a microscopy or something. A cartoon showing a macrophage chasing some foreign particles. Right? These particles are shown here as yellow spheres with something sticking out from them, but closer to the front you see also egg-shaped substances with something that is sticking out of them. And these substances, just use your imagination, some of them may be not colored yellow, they can be colored green or blue or red. It does not matter for the macrophage as long as the substance is foreign, comes from outside, not me, right? Or it is modified me, like a cell that undergoes cancerous transformation. The macrophage just eats it. Oh, see, it is shown here like a shark which opens its mouth and swallows these foreign particles. Doesn't matter what. There were some experiments where macrophages were cultured in sterile plates with no foreign invaders whatsoever, and they were fed by sterilized beads of latex, right? It's absolutely not a microorganism, but macrophages ate them nonetheless. They swallowed these pieces of, well, latex because it's foreign to them. It's not me. It's not part of my own host, my own favorite organism to which I belong. It's foreign, so I'll better eat it. I don't think that they felt very well after they ate pieces of latex, but they do. They just eat everything that is foreign. Now, the bottom part of this picture, it shows you this green thing that stands in front of the counter. It's shaped like letter Y. It's an antibody. And antibodies are specific. They're part of this of specific immunity, specific immune responses, specific to particular antigen. And you see this person behind the counter sells different pieces of whatever. I don't know. They are actually foreign invaders antigens, but some of them have this triangle shape, others have rounded shape, yet others are rectangular, etc. And this salesperson behind the counter kind of offers all these different kinds of antigens to the antibody. But it just so happens that only this triangular piece of something, an antigen, fits into the antibody. Right? All the rounded do not fit. Rectangular things do not fit. Only this triangular, because this antibody has some like grooves that are and depressions which accommodate this triangular thing. So we say that antibody acts against the unique matching antigen. Antigen is like a key, and antibody is like a lock with keyhole, which will accommodate only particular key, not all keys, God forbid. Imagine your locks at home can accommodate all kinds of keys, you will definitely change these locks, right? They don't serve you well. So antibodies are like perfect locks. They accommodate one particular antigen. Uh, a little bit more about antibodies. They are protein molecules. Please remember, they are produced by B lymphocytes, by B cells, but themselves are not, they are not cells. They are molecules of protein. Such a common mistake that people say and write sometimes that the immune system produces immune cells called antibodies, and antibodies fight against foreign. No, they are not cells. They are molecules with particular structure. Like all proteins, they consist of amino acids. The specific sequence of these amino acids in a larger part of antibody molecule is the same, but there are parts of antibody molecules which have varying sequence of amino acids, so that makes these antibodies specific only to particular antigen. Uh, B cell, when it makes them, it puts them on its surface. And so in the picture below, there is a B lymphocyte, and you see antibodies are sticking from it. And on previous slides, you saw a B lymphocyte with an antibody molecule sticking out like an antenna. But these molecules can also be secreted. You see, there is in this picture below, there are antibody molecules that are just floating. They're not attached to any cell. So the B cell detached them from the surface and they now can circulate in blood, in other body fluids. There are antibodies, for example, in sweat. There is a lot of antibodies in mother's milk, by the way, which is one of the reasons why breastfeeding is so wonderful, because it supplies the baby with some important antibodies which can defend the antibody. So it depends the baby against invaders. Then, like I said already, the antibodies have parts which fit certain antigens, like a keyhole in a lock fits only certain key. And one more thing, you may hear sometimes this word is used, immunoglobulins. Why immunoglobulins? Because biochemically, antibodies belong to a class of proteins called globulins. If you take an organic chemistry or biochemistry class, they will explain in detail what this class is, what are its properties. But you just right now just remember that antibodies biochemically are globulins, but they are parts of the immune system. So one synonym for antibodies is immunoglobulins, shortened as IG. And in humans, there are five classes, five kinds of these IG molecules, termed with different capital letters of the Latin alphabet. Like there are IgE antibodies which specialize in allergic reactions. They are not very good antibodies for us because they react not as much with foreign invaders, or pathogens, as with particles of, say, pollen or house dust, etc. They are a class of antibodies that cause allergies. Other classes are not like that. IgM, IgG, they are very good. They are useful. They react with other, not with different antigens, including antigens of pathogenic microorganisms. Now, clonal selection and immune memory, very important part of your learning about the specific immune system. Lymphocytes, well, like we said, lymphocytes have very, very different specificities. Right? One lymphocyte is specific to tetanus toxin, another to diphtheria toxin, yet another to measles virus, yet another to influenza virus, etc., etc. They don't share their responsibilities. So, how many different specificities are there in you? The different specificities are shown in the upper
level of this picture, and you see 3 lymphocytes. But in reality, do you know how many different specificities of lymphocytes you have in your organism? I'll surprise you. A trillion. A trillion. It's amazing, but many laboratories employed different methods to estimate this number of specificities, and they all came to this huge, astronomic number of individual specificities, or individual shapes of these, the so-called antigen receptors. Some of them are antibodies, some of them are on T cells, so they're not antibodies, but in any way they are specific to antigen. And so there are, you at any moment of time, regardless actually of whether you are infected or not infected, whether some foreign intruder bothers you or not, doesn't matter. At any given moment of your time, from your embryonic development till the day you die, you have 1 trillion of different specificities of lymphocytes. Then, what happens when antigen arrives? See, at the top of this picture, you see 2 purple-colored spots, and they are called antigen molecules. They fit into only one block, right? Into one selected specificity of antigen receptors. So the lymphocyte on the left is not selected by the antigen. Lymphocyte on the right, again, it's not selected. The lymphocyte, receptors for antigen on these lymphocytes on the left and on the right do not fit the antigen. So antigen ignores them, and if not any other antigen selects them, they will die in approximately between 2 and 3 weeks. They don't live too long. And then, but if the antigen molecule fits the antigen, what happens? The lymphocyte that carries these receptors fitting the antigen is activated. There are many different biochemical reactions that proceed in this lymphocyte. And the pinnacle of this change during activation is ability to divide. See, lymphocytes not selected by antigen do not divide. But a lymphocyte that has been selected by antigen divides very actively and produces a clone which is growing. It's 10, 20, 40, 80, 1,000, 2,000, million, 2 million, 10 million, 100 million cells. Some clones can reach very large size. They really keep growing and growing and growing. When your immune system responds to the so-called Epstein-Barr virus, which causes infectious mononucleosis, you have huge clones of lymphocytes in your blood that reach an immense number of cells in them. And then what happens? So these divided cells, the cells that grew, sizable clone, all of them react to the antigen. If one lymphocyte reacts to the antigen, nothing will really happen. But if a million of cells react to antigen, like secrete antibodies or do something else, and it does matter. It means that you develop an immune reaction. Now, what also happens, see here, you see this clone in the middle, and then 2 arrows pointing in different directions. Within a particular clone, some cells, you see them on the right, under the arrow that leads to the lower right corner of the slide. You see cells that immediately, well, almost immediately, it's not really instantaneously, but very, very quickly, relatively, respond to the antigen. They produce antibodies. See, they secrete antibodies. They differentiate, if they're B lymphocytes, they differentiate into so-called plasma cells, which secrete about between 1 and 2,000 antibody molecules per cell per second. So an immense number of antibodies. But there's another part
in the clone, and you see it in the lower left corner. It says clone of memory cells. These are very peculiar cells. They respond to antigen, but not right away. When they encounter antigen for the first time, they divide and acquire a huge, immense, tremendously long lifespan. That's the paramount feature of memory cells. They live long. So the plasma cells live a couple of weeks. Imagine a cell that works so hard, secreting 2,000 molecules per second. Every second it has to throw out 2,000 antibody molecules. It becomes exhausted. It doesn't live very long. It dies pretty soon. But memory cells live for a year, 2, 10, 20 years. Some clones of memory cells live as long as you live, right? So when you encounter certain antigens, you have response to these antigens, and you have forming of memory cells which wait until this same antigen comes another time. Okay, the second visit of the same antigen causes this explosive reaction. As you see, during the response to the antigen that appears for the first time, the specific lymphocyte must divide, form a clone, and it takes days. Right? It doesn't take seconds or minutes or hours even. It takes, on average, between 9 and 11 days, and usually more than a week. Right? During this week, actually, antigen, imagine that this antigen is on some harmful bacterium. It has free reign, upper hand. Only when this clone of a reacting lymphocytes reaches large size, then this antigen can be harmed. But not so during the second invasion of the antigen, because clones of memory cells do not have to divide further. Right? There are already millions of cells in these clones. So they react all at once, very quickly and with very strong, powerful response. And I'll tell you right away, we'll talk about this more further in lecture. But the action of vaccines is actually based on this phenomenon of memory cells. Good vaccines are those that form good immune memory. Right? Vaccine is an antigen. It is an antigen associated with a microorganism that had been weakened, chemically, for example, chemically treated, killed. It cannot cause infectious disease, but it carries the same antigen as harmful microorganisms. So when you're vaccinated, your immune system responds to the vaccine. You don't usually you don't notice this primary response. Sometimes you do. You see reddening and some inflammatory reaction, but usually you don't even notice it. And then also, one part of response to the vaccine is forming of the clones of memory cells. Then, when you, God forbid, are attacked with harmful microorganism, which carries the same antigen that your immune system has already seen on vaccines, on a vaccine, it responds now to the harmful, to the pathogenic, to the dangerous microorganism, destroying it, and sometimes the reaction is so fast that the intruder, some harmful bacterium, is destroyed even before this bacterium causes symptoms of disease. Bacteria that cause diphtheria, they produce toxins which glue together cells of the respiratory tract, and this suffocates the patient. But if you're vaccinated, you have clones of memory cells specific to this diphtheria antigen. When diphtheria attacks you, Corynebacterium that causes diphtheria, attacks you, the reaction of memory cells is so fast and so strong that you don't even develop symptoms.
This bacterial toxin, diphtheria toxin, does not have enough time to glue together cells of your respiratory tract. All the molecules of this toxin are neutralized within 1 day or maybe 2 days, not more than that, very fast and very strongly. Okay. Uh, you know, subsets of lymphocytes. Lymphocytes are not all the same. You already heard that there are B cells that produce antibodies and there are T cells that do not. But T cells exist as three separate subsets. On the left, you see the so-called CD8 T cells. CD8 is a special molecule expressed on the surface of some T cells. They are killers, like Bill in this movie, Kill Bill. They are killers. They're professional killers. They don't do anything else but kill. When they recognize, for example, parts of a virus which lives inside a cell, divides inside a cell, they see these markers of the virus, and how do they respond to this? They kill the cell, annihilate it. They bombard the cell with special toxic, harmful substances that break the membrane of the cell that carries the virus and break the nucleus, break even the DNA of the cell that harbors the virus. So there's no chance. There's no chance for this virus to get away from killer CD8 T cells. And next from the left is a B cell. You already know about B cells. Then next, third from the left is a regulatory T cell. Regulatory T cells are policemen which work like, well, some pretty nasty cops. Their function is to suppress, to put down the rebellion. They watch out for excessive immune responses. And when they see this continuing immune response that does not really serve any useful service anymore, they suppress the division of cells, they suppress the production of antibodies, they suppress production of these signaling cytokines, etc. And first from the right, or fourth from the left, CD4 T cell. It is we call it helper. CD4 cells are helpers. They produce the signal molecules called cytokines. You saw them produced by macrophages in the slide that showed you inflammation, but most cytokines are actually produced by CD4 T cells, and these cytokines regulate immunity. They have a number of different functions. They can stimulate CD8 T cells, they can inhibit CD8 T cells, they can stimulate B cells and antibody production, or they can inhibit B cells and antibody production. So they basically optimize the body defense. They make the immune response just fitting its purpose. One example, if you are, God forbid, infected with bacterium that causes tuberculosis, it's intracellular, and so it only lives inside cells. Antibodies, B lymphocytes producing antibodies. Are they useful? Can they contain these bacteria? No, they don't crawl inside cells of the respiratory tract, cells of the lung. Antibodies remain outside of cells. They cannot cross this barrier. And so the bacterium inside your cells of the respiratory tract continues to divide and produces, may cause the cell to burst, or may cause granuloma and necrosis of the tissue of the lung. And here we have CD4 positive T cells, or CD4 T cells, which in this case inhibit B cells, inhibit the production of antibodies. You see, they perceive it as a waste. The tuberculosis bacterium lives inside cells, so the antibodies are not efficient in the first case. Why allow B cells to waste energy and time producing antibodies? But the CD4 T cells produce cytokines that stimulate CD8 T cells, right? So many of us encounter this bacterium that causes tuberculosis, but most of us do not get ill with tuberculosis because CD4 T cells actually stimulate some T cell responses that do not allow the infection to develop. Okay, so that's lymphocyte subsets for you. Then, a very important thing and subject that I want you to really have some idea. Vaccines, vaccination. What is a vaccine? It says here, a vaccine is a biological preparation that improves immunity to a particular disease. Right, but I would say more specifically, a vaccine is an antigen. Right? It's an antigen which was rendered harmless. Right? It's like, there are cats with big claws, and if they are aggressive, if they scratch you, sometimes veterinarians make these claws sort of dull, right? So the cat still has something on its paws, but it cannot harm you because it was made harmless. So vaccines are harmless variants of bacteria, viruses, toxins, etc. They stimulate immunity. They cause the forming of the immune memory, but they do not cause infectious disease themselves. A vaccine is a word derived from usage of cowpox. Cow means in Latin vacca, or vacca means, it's not, I would change the sentence. Vacca in Latin means cow. Vaccine is a Latin word which is derived from vacca and vacca, and basically it means from cows. The portrait of a British physician, Jenner, who first isolated material from cows ill with cowpox and injected it into the shoulder of a boy who became immune to real human smallpox. So the vaccine, material that Jenner took from cows actually helped the boy to remain healthy when many other people in his village succumbed to real human, not cow, but human smallpox. Now, it says it contains an agent that resembles a disease-causing microorganism, I would add, or its parts, like toxins, and is often made from weakened or killed forms of the microbe. Then the agent stimulates the body's immune system to recognize the foreign antigens and destroy it. I would, as an immunologist, I would better say that the agent or the vaccine stimulates not just the body's immune system, but more specifically immune memory to a certain particular antigen. For example, if you are vaccinated with standard DTP vaccine, it has toxins from diphtheria, from tetanus, and from pertussis, right? Then you become immune to these diseases, diphtheria, tetanus, and pertussis, because your immune system has formed immune memory to toxins of bacteria that cause diphtheria or tetanus or pertussis. And so if real bacteria, not the vaccine, but real, alive and well and happy, proliferating bacteria attack you, for memory cells, it doesn't matter whether it is vaccine or it is the population of real harmful, dangerous bacterial cells. So these lymphocytes from memory cell clones attack and destroy and neutralize toxins, and it happens very fast so that you don't even notice any symptoms of tetanus or diphtheria or pertussis. Okay, I will just add to this one thing that sometimes people have very strange notions. Some people have adverse reactions to the vaccine against influenza virus, and they say, okay, well, Why would I be vaccinated? This vaccine makes me ill. It actually is influenza. It causes influenza in me because when I was vaccinated, I became uh, uh, tired and fatigued, and I lost my appetite, and I had some pain in the muscles and high temperature, etc. So I got influenza from the flu vaccine. This is impossible because actually the current influenza vaccines do not contain influenza viruses. It may be strange, but it is true. They don't contain even killed influenza viruses. They are mixtures of proteins extracted from viruses. So they cannot, it's not a living object. It cannot infect your cells and cause influenza. What you have is simply adverse reaction, which will come and go away. It doesn't really make much difference, but you will be protected, likely, not absolutely, but there is some chance that you'll be better protected against influenza virus. And especially children, they are at risk for influenza. They should be vaccinated, and elderly people also should be vaccinated. Young adults, young people, optional, because vaccine is not really 100% efficient. Its efficiency varies from 50%, from 25% actually to 50%. So if you are vaccinated, you're not still completely immune from completely not susceptible to the influenza virus, but you have a better chance not to get influenza during this cold period when influenza risk is. Then what can go wrong? I will be very brief. Some immune reactions are actually harmful. Allergies are a form of harmful immune reaction. You know what allergies are, right? Hardly any one of us is not allergic to anything. I thought of myself as not allergic for anything, but actually, if I recall my childhood when my grandmother gave me strawberries, and I refused to eat them because I said they burn my palate and my tongue. And of course, my grandmother, grandmothers are great specialists in everything. She said, "You're just spoiled because there are so many children who do not have strawberries and they want to have strawberries. You have strawberries and you don't eat them." But it's quite really, now I realize, I understand the time I showed symptoms of allergy to strawberries. If a person is allergic to some allergen, dust, pollen, components of some food, etc., then the allergen plays the role of an antigen that triggers production by B cells of antibodies of the so-called IgE class. I mentioned them earlier. Those are specialized antibodies. They have virtually no useful function. They don't target bacteria or viruses. They react with these allergens. They bind these allergens. And see here, you see a plasma cell to the left, which is it's basically a differentiated B cell that's specialized on secretion of antibody, not as much showing it on the surface as kicking it out, secreting it, make it circulate in body fluids. And unfortunately, fortunately, because these cells also have useful functions, but also unfortunately, we have some cells that trap or capture these IgE antibodies. They have special receptor for IgE antibodies. And you see here, antibodies are sticking out from a cell that is called mast cell, right? Or also from a cell that is called basophil. It's not shown here. There are two kinds of cells that can capture IgE, mast cells and basophils. Then it doesn't mean that they produce these. Only B lymphocytes can produce antibodies. Mast cell cannot produce even one molecule of antibody. So basophil, same thing. But they capture, trap IgE antibodies secreted by plasma cells. And then suddenly You are attacked by the same allergen again. First encounter with allergen, usually you don't notice. But second encounter by allergen, and what you see here is mast cells, also basophils not shown in the slide, but they act similarly, throw out special granules. It says here, mast cells degranulate. And these granules have substances that cause allergies. These substances cause skin rashes, swelling of the face, sneezing, sometimes constriction of the bronchi, asthma. These substances vary. Some of them are less harmful than others. For example, histamine is one of the mediators of allergy, but you have some itching or something, and you take Benadryl, which is an antihistamine drug, and it goes away. And so it's relatively easy to fight, to make go away with medication. But other substances normally contained within granules and thrown out from the granules of the mast cells and basophils during the allergic reaction have very dangerous substances. Like, for example, there are substances called leukotrienes. You may not remember that, but just remember that some substance can cause constriction of the bronchi. Asthma attack is actually also a form of allergic reaction. What it leads to, if your bronchi are constricted, you can still inhale, because even if bronchi are narrow, the diaphragm is powerful enough to drive air into the lungs. But recall respiratory system, what's exhalation? It's simply relaxation of the diaphragm. And it's not enough to expel air from the lungs because the bronchi are too narrow, too constricted. This is what asthma is. If you know some people with asthma, they will tell you that my problem is that when I have my asthma attack, I can inhale, but I cannot exhale. It's such a terrible feeling. It's actually the chest cavity inflates, and the air just doesn't get out, and you feel like you're strangled, suffocated, hung, or whatever. Like you're strangled by some evil force. In this case, you need to take some bronchodilators, substances that dilate bronchi, like adrenaline, ephedrine, and others. They dilate bronchi. But to address the root of the problem, of course, you need substances that inhibit the production of IgE antibodies. Now, what else can go wrong? Autoimmune diseases, again, are kind of immune reactions that are very harmful. You know, not all immunity is helpful, protective. There are some immune reactions that are terribly harmful. Allergies, we mentioned. Now, autoimmune diseases. What does this term mean? It means that the immune system goes nuts, literally, because of something that happens during maturation of precursors of immune cells in bone marrow or in the thymus, or functioning of immune cells in the periphery, in the lymph nodes, in the spleen, etc. Something goes wrong, and the result of this something going wrong is that your immune cells stop protecting you. They stop attacking substances that invade you. Instead, they attack you. They attack tissues, cells, substances of their own host. There is a lot of them, actually, many diseases, and some very common diseases are autoimmune in origin. You definitely heard about rheumatoid arthritis, right? When small joints hurt, like joints of the fingers, phalanxes of the fingers, hurt so much that people have to take strong painkillers because otherwise they feel miserable. They cannot really move their fingers. They cannot utilize these articulations between phalanxes. That's because the immune system attacks something that is on the synovial surface of small joints in fingers and toes, etc. Then lupus, full name systemic lupus erythematosus. You heard about this disease, I'm sure. It's one of its features is that it is predominantly female. The ratio of females to males ill with lupus is 10 to 1. Males can get ill with lupus, but rarely. And for women, it's more prevalent, more frequent. The lupus manifests as skin rashes, and especially why is it called lupus? Because it causes this butterfly-shaped skin rash around the nostrils. And some people, I don't know, I'm not sure why, but some people in old times, they saw some similarity between the face of a woman ill with this disease and face of a wolf. And lupus is Latin for wolf. But it's much more dangerous than that. It doesn't only affect skin, it affects internal organs, especially kidneys. Lupus nephritis, very severe. Women who have lupus have to get very strong immunosuppressive therapy because otherwise they have a big chance to die of kidney failure. Type 1 sugar diabetes, also autoimmune disease. We talked about type 2 diabetes. It's actually a disease of a lifestyle. Type 2 is caused by abusing sugar, eating unhealthy, eating sugary foods, fatty foods, and sitting around, not exercising. That's what causes the resistance of tissues to insulin. Insulin in type 2 diabetics is normal or even higher than normal. But in type 1 diabetes, it's a totally different thing. Insulin is just not there because the patient's own immune system destroys the cells in the patient's pancreas, which normally produce insulin. Right? So it's called insulin-dependent diabetes, and it's a clear-cut autoimmune disease. Multiple sclerosis, it's a disease of the nervous system, and it is caused mostly by T lymphocytes, not B cells, not antibodies, but autoreactive T cells that destroy myelin shields in the nervous system, make neurons bare. They rid neurons from normal myelin covering. And it manifests as difficulties in speech, articulation, difficulties in walking, difficulties in physical exercise, etc. Nowadays, actually, patients with multiple sclerosis are, when they receive enough immunosuppressive therapy, they look kind of like healthy people, but they must remain on these immunosuppressive drugs. Graves' disease, that's the disease that manifests as hyperfunction of the thyroid gland. If you have ever seen people with Graves' disease, you will never forget how they look. They're very thin. They don't get weight, right? They don't accumulate fats and glycogens, anything in their body. Their metabolism is very high, too high, pathologically high. They have a certain stare. Their eyes are kind of staring at you. It's not because they want to stare at you, but it's because of the paralysis of certain muscles that are keeping the eyeballs in the eye sockets. So these people suffer tremendously. They need either surgery. Surgeons can eliminate part of the thyroid gland, or they need radioactive therapy. Some radioactive isotopes, iodine-131, does basically the same thing as the surgeon's knife. These isotopes eliminate parts of the thyroid gland, and they help people with Graves' disease, but they don't liquidate the disease because autoimmunity remains there. Sjogren's syndrome, you might know, and it's predominantly female disease, and it manifests as dryness of surfaces, dry skin, dry eyes, dry mouth. Again, women, people, patients need immunosuppressive therapy. Autoimmune hemolytic anemia, it's when a patient may eat normally, enough iron, enough hemoglobin apparently, but really not enough hemoglobin because the numbers of red blood cells are going down, 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 down. It's not because the hemoglobin cannot be synthesized, but because antibodies of this person destroy this person's own red blood cells, which is anemia, hypoxia, yellowness of the skin, yellowness of the white of the eye, because dead red blood cells they lose hemoglobin. The hemoglobin out of red blood cells turns into the so-called bilirubin, which has a yellow color. So these people have jaundice usually. So again, they need immunosuppressive therapy. It's a pity that we cannot really treat right now the cause of autoimmune diseases, because we do know something about genes that are involved, about biochemical reactions that are involved in autoimmunity, but we do not still have means to treat patients with autoimmune diseases so that we eliminate the self-reacting immune cells and self-reacting antibodies. We cannot do that. We just can suppress the entire immune system, making this person happier for a while, but prone to infections, by the way. Immunosuppressive therapy is not indifferent, makes you more susceptible to infections. Then, and immunodeficiencies. Immunodeficiencies are pretty common, actually. They're not rare. They are diseases when the immune system has no resources to mount an efficient response to infections. You see, it's different from autoimmune diseases. In autoimmune diseases, there are resources, but they are targeting the wrong thing. And in immunodeficiencies, people have an immune system which has immune cells and antibodies, cytokines, etc., targeting infectious agents, microbes, but not enough of them. These patients have low B lymphocyte count, sometimes zero B lymphocytes, low T cell count, low T and B cell count, and other manifestations of insufficient immune responses. The immunodeficiencies can be primary or genetic. So some people are just born with wrong genes. For example, people with the so-called Bruton agammaglobulinemia, people with severe combined immunodeficiency, which is pictured on the slide. See this poor little girl. She looks pretty healthy, but she's held in a bubble, basically in an incubator. She's isolated from the environment because if she is let out from this bubble, some microorganism will attack her and she might die because she just does not have enough resources to fight infectious disease. Or immunodeficiencies can be acquired. And you certainly know acquired immunodeficiency syndrome, or AIDS. It's a disease caused by the virus, HIV. It is contracted either sexually or by body fluid exchange during sharing needles, or by passing the virus from the mother to her fetus. People can be born with AIDS, but we still do not consider it primary because it's not bad genes. It's just that the transmission happened in the part of the person's life when the person was still inside the mother's womb. And you know, I do not want to go into depth, but you know that right now there is no vaccine against AIDS. There are medications that can contain the virus, right? And so if a person is tested positive for HIV, this person must receive treatment, must receive drugs that slow down the viral replication. And this prolongs the person's life, and it also lessens the possibility of transmission of active disease-causing virus to other people. But it's still it's very difficult to get rid of viruses like HIV. It will still remain somewhere hidden in the body, so the HIV might relapse, and if it is a full-blown AIDS, it's usually death of so-called infections by organisms microorganisms that normally live in us but don't do us any harm. Opportunistic infections. Normally, for example, there are yeasts they live in the lung of human beings between alveoli. And the immune system keeps them under check so they do not proliferate seriously. But if the immune system is gone, because HIV actually destroys the immune system entirely, then this yeast proliferates like crazy and replaces the normal tissue of the lung. So the lungs of AIDS patients become like sacs that are filled with yeast instead of sacs that are filled with alveoli, which provide gas exchange, etc. So the person's lungs are now useless, and this person must be put on the ventilator, on the artificial breathing apparatus, etc. Or many of the full-blown AIDS patients also die of tumors because, again, the immune system is gone. There's nothing to contain tumors. And tumors, especially capillary sarcoma and lymphomas, are death warrant for AIDS patients, extremely dangerous. And one last uh, slide about, or not last, there will be one more, sorry. Immune rejection, previous to last. Immune rejection. You heard about immune rejection, right? It's the situation when something is transplanted. There are no transplants in nature, but we humans invented transplants. And transplants are ideally efficient, successful, only if tissues or organs are transplanted from one identical twin to another. Identical twins are genetically identical. So there is no need to match the donor of the transplant and the recipient of the transplant. They are like Xerox copies of each other. Biologically, they are like absolutely genetically the same. And but in all other cases, when you have genetically different individuals, right, then either the uh, recipient will reject the transplant, or the immune system of the recipient will try to get rid of the transplant, destroy it. Or in some cases, especially during bone marrow transplantation, the immune system of the donor, transplanted bone marrow from a donor, will try to destroy the tissues of the recipient, called graft versus host disease. Both situations are very unpleasant, and it is shown, it's a cartoon that shows different kinds of immune cells and a little heart which is transplanted into this probably non-matching individual, and the immune system of this recipient tells the heart, get lost, get lost. And so rejections are still happening. It's a very sad outcome. Kidneys get rejected, hearts get rejected. Uh, Bone marrows are not frequently rejected, but they destroy tissues of the recipient. For example, uh, immune cells that are growing from the donor's bone marrow can destroy the host's liver or the host's skin. There are some patients with severe graft versus host disease. they look like their skin was scratched from. They're like being flawed, right? Flaw is the word to remove skin. They look horrible. They need to receive also immunosuppressive therapy, in this case to suppress the immune system of the donor, which attacks their tissue. And also, but we don't actually have to worry so much about rejection if we carefully match a donor and a recipient. If a donor and a recipient are at least partially genetically matched or compatible, then there is a higher chance of successful transplant. For identical twins, it's the 100% chance of success unless a surgeon did not cut their artery or something. If the surgery was done right, they will not reject the transplant. For mothers and children, there is a chance that the child will reject the mother's tissue, or the mother will reject the child's tissue because they're not genetically identical. But it's a lower, this chance is lower than if we just take a donor from the street and transplant something from this random donor to the recipient. And, or from brother to brother, from sister to sister, that actually works better, again, than just random transplant. But immunosuppressive therapy still, it has to be considered, and in very many cases it must be given. Right. So the life of recipients of transplants, just like the life of people with autoimmune diseases and immunodeficiencies, it's not a very happy life, and unfortunately it has to be, people must be on medication. And finally, this is really the final slide, immunity and stress. It's been shown that good mood, happiness, stimulate immunity. When you are in a good mood, when you are happy, your endocrine system produces some hormones that stimulate immunity. Your neurons, nerve cells, also produce some neurons that stimulate immunity. And so your antibodies are produced better, your T cells react against antigens better, etc. Macrophages, dendritic cells, they're all improving. People who are chronically stressed have a greater chance to suffer from infectious diseases because, again, stress always is accompanied by production of some substances that suppress immunity. One simple example, glucocorticoids, you know, cortisol, hydrocortisone, those are stress related substances, and they deeply, deeply suppress immunity. So people who are chronically stressed, people who are unhappy because of whatever, people who have huge demands, people who are always sad or angry because they think that they did not receive enough from others, from the society, etc., etc., they are more likely to get infectious diseases. And I personally know examples of people who, one of some of my relatives, unfortunately, they always get cold and flu every year, or strep throat, or common cold, or influenza, or some rhinitis, or some immune attack on their joints, etc. So they are always not just they're always unhappy in part because they're ill, and they're ill in big part because they're unhappy. So be happy. Teach yourself to appreciate life. Be happy just because there are so many wonderful things around you. And do not expect people to give you everything you need. It's not gonna happen. There are some things for which you have to work. And don't get upset, for example, with a teacher who gave you a D or an F on a quiz or on an exam because there are certain things that they don't really depend so much on the things outside of you. They happen because something that is inside of you, your laziness, your um, uh, propensity to be distracted by other things, and simply not studying, right? But when you study, you learn, and when you learn, your grades improve, and when your grades improve, you become like these people pictured here. So just learn, just study and learn, and read and watch good movies and go on hikes and see wonders of nature and read good books. It's really reading of a great, wonderful book is something that can make any person happy because some books are so wonderfully written that you just get pleasure by reading them. You remember them. They live in you and they make you happy. So please be happy. Eliminate non-essential worries. I'm not telling you don't worry about anything. Yeah, well, if you have an exam tomorrow, you do worry, but don't worry, for example, that I don't know, like there's one German fairy tale called Poor Elsa. Elsa was worried always, and once there were visitors, and she started to cry, and they asked her why was she crying, why was she unhappy, and she said, "I am worried because if my father, who is he works in the woods. He's a woodcutter. He always walks with his axe near his belt. And if he forgets this axe in the cellar, and somebody picks the cellar and tries to throw it out, but misses the exit from the cellar, so that the axe actually hits the ceiling of the cellar and hangs from there, and then I have you guys will go away, but I will have soon other visitors, and one of them will go to that cellar, and the axe will fall on that person's head. So I'm already worried, and I'm already unhappy. So don't be like poor Elsa. Eliminate unnecessary worries. Don't get worried about things that you do not control, but be pursuant, be tireless pursuing the goals that you can achieve, right? And you will be happy, and your immune system will improve. You will not get cold every cold season. Okay, thank you so much for listening, and goodbye until the next lecture. Best wishes to all of you.